today we are very excited to talk to Dr. Lulu Jiang of Boston University Medical School. She's a research assistant professor in the Department of Pharmacology and Experimental Therapeutics. And she's going to talk today about stress granules, deciphering the connections to neurodegeneration. And you might wonder why this might be pertinent to type 1 diabetes research. But if we look at some of the literature, there is some work that describes saturated fatty acids entrapping PDX1 in stress granules and impeding islet beta cell function. That came out in Diabetology, May 2021 from Lulu et al. And that group is, in Nanjing, China. But they are, this kind of is involved, this whole process is involved in type 2 diabetes, but it does ask whether or not this kind of process in stress granules may also be contributing to problems in the beta cell functionality. So just a little bit about Dr. Jiang. She was a postdoc fellow in the Laboratory of Neurodegeneration, mentored by Dr. Benjamin Wallison. Her previous work demonstrated that propagated tau oligomers, but not tau fibrils, induced pathological changes in RNA metabolism and neurodegeneration. And upon that, Dr. Jiang developed an optogenetic system for controlling tau aggregation, which brought in a seminal discovery that tau oligomerization drives the accumulation of RNA-binding protein HNRNPA2B1 and N6-methyladenosine modified transcripts in the aggregated tau complex. She also is the lead on the generation of human 3D iPSCM/ASTAD based models of AD for the exploration of molecular mechanism and drug screening. Her research interests include protein-protein and protein-RNA interactions in AD and related dementia. Secondarily, molecular mechanism of tau prion like propagation in break stages of AD, neuron-glia interactions in pathogenesis of AD and PD, and as well as iPSC-induced 3D brain organoid models of neurodegenerative diseases and precision medicine. And finally, she's very interested in developing novel therapeutics for neurodegenerative diseases. Thank you very much, Dr. Jiang, for joining us. And we look forward to your talk.
Thank you very much for the introduction, Monica. It's my great pleasure to have this opportunity to introduce our research and also like to the Sugar Science Foundation. I think it's very helpful to and to think about this in a broad view of research and studies. Like you introduced, our group focused on the studies on stress granules to decipher the connections to neurodegeneration and to explore novel therapeutic strategies. So first, we introduce what is stress granules. Stress granules are dense aggregations in the cytosol composed of proteins and RNAs that appear when the cells is under stress. And how it links to neurodegenerative diseases. As we all have some knowledge about neurodegeneration. It's a group of disease with a common feature of progressive neuronal loss. Most of the, including Alzheimer's disease, frontotemporal dementia, Parkinson's disease, Huntington's disease, etc. Most of these neurodegenerative diseases are not curable. There's no therapeutic strategies that can reverse the disease or cure the disease. The only medications available nowadays is to control the symptoms, but that is only temporarily. So it's important to find the fundamental mechanisms related to this disease and to treat them from the beginning and from the early stage. So stress granules is a very important component in the neurons that are degenerating, especially in the neurons with tau pathology or amyloid pathology. In the neurodegenerative diseases, an important feature of this disease is that they have the misfolding of proteins. They have different proteins involved, but these proteins are all misfolded. For example, tau protein is in Alzheimer's disease, in frontotemporal dementia, and in ALS, that is TDP43, and in Parkinson's disease, that is called alpha-synuclein. the different proteins, but they have the common feature of misfolding. And when this protein get misfolded, they will recruit other proteins and RNAs in the complex that form a membraneless dense aggregation. So we call it stress granules. So in these stress granules, when it's acute process and temporary, they are reversible. So the neurons have this process to help themselves to preserve the RNAs and proteins. But once this process become chronic and become irreversible, they will develop into disease. So that is the beginning of the initiation of the disease, and also the fundamental questions we think that's important to know and important for the therapeutics in the future. So that's why we focus on the stress granules in the lab. So like I introduced in the stress granules composed of proteins and RNAs, and also the misfolding aggregates. And these RNA-binding proteins is a group of proteins that is modify or help the translocation or the transportation of RNAs. The RNAs is modified in the nucleus, and once they are mature, they needed to be transported to the cytosol, to the cell body, to be functional. So in the physiological conditions, this is worry, everything is under like in the regular process, but in disease, this process is dysregulated, and then these aggregates deposit in the cells and become irreversible complex that can influence the normal function of neurons. And this is an overview of how the stress granules look like in the physiological conditions, the blue side, and also in disease conditions, the pink side. So under normal, in the normal conditions, they form and they aggregate and disaggregate, that's kind of dynamic process. But in disease, the dynamic is disrupted. These aggregates accumulate and accumulate. They not go back to the normal conditions. So we hypothesize these persistent stress granules related to the neurodegeneration. The RNA-binding proteins in this process is multiple. So until now, the related RNA-binding proteins we found include TRA1, TDP43, and also G38P1. Those are already found. But because of the complex contains hundreds of proteins and also RNAs, so we still don't know which is the key component and how to disrupt the process of aggregation. This is the overview of how this complex forms and become irreversible. There's a core protein called tau. It's a microtubule binding protein, and in normal conditions is the cargo to transport proteins and RNAs from nucleus to the cytosol and from cytosol to the neuronal axons. But under disease conditions, this protein is stuck in the soma, in the cell body of neurons, and it recruits other RNA-binding proteins and RNAs from this complex. So nowadays, a lot of studies focus on tau protein, especially Alzheimer's disease. In the AD field, amyloid plaques and neurofibrillary tau tangles are the two main features of AD. In the past 30 years, a few decades ago, everybody thinks amyloid is the key to cure the disease. So a lot of drugs and also a lot of clinical trials focus on how to resolve the amyloid plaques, but they all failed. So in the recent 10 years, people started to look at tau because people found the tau aggregation is highly correlated to the cognitive decline. So that maybe is the clue to treatment. So, but unfortunately, the cellular mechanisms that regulate tau or amyloid aggregation and its cytotoxicity is still unknown. In our lab, we hypothesize that the core protein tau recruit RNA-binding proteins and also change the RNA metabolism. And like I mentioned, most of the neurodegenerative disease have the is progressive and incurable, and it's very economic burden, and also a big hit to the families with patients. And not only Alzheimer's disease, but also other neurodegenerative disease, including chronic traumatic encephalopathy, PSP, cortical basal degeneration, frontotemporal dementia, and Pick's disease. These all have the same feature of tau misfolding. That is the core protein related to these different neurodegenerative diseases. So the tau protein is really important, and it's we tried many years to understand how tau was dysregulated and how there was misfolded. In the field, there's a common knowledge that tau at the beginning is a microtubule binding protein and located in the axons. It's very enriched in axons, but not in other compartments of the neuron. But under disease, this tau protein is highly phosphorylated. Once they are phosphorylated, they dissociate from the microtubule and they become floating and freely moving around. So once they move to the cell body, the soma area of the neuron, they will capture other proteins or RNAs and accumulate into an aggregate. At the beginning, they are small aggregates. We call it tau oligomers. That's a few replicates of the tau proteins. When they become accumulated more and more, they will become large fibril tangles. We call it tau fibrils. People think because in the postmortem brain tissue, human brain tissue, they always find these tau fibrils. It's a large tangles. They think this is important, but after a few years of work to compare the toxicity of tau fibrils, they found fibrils actually is not that toxic. And also our lab, we try to understand the dynamics of tau misfolding, and we designed experiment from beginning to the end in the animal model, trying to understand the monomers of tau, how the structure and the interaction with monomers tau, oligomers tau, and fibril tau. So in our study in a tau pathology mouse, we found these kind of interactions with tau pathology, including the RNA metabolism and also mitochondria, and also to the synaptic and vesicles, also the cytoskeleton. And this part, the most important part, RNA metabolism, caused attention because these proteins, for example, TIA, ERF4, and also HRMPF, EWSR1, this is a group of protein called RNA-binding proteins. These are the most important proteins for stress granules formation. So we thought, oh, maybe this is the key to understand how the tau oligomers induce the dysregulation of RNA, the dysregulation of protein translation, and also later to cell death. So to understand that, we focus on the interaction of proteins with tau and RNA binding proteins. So here shows how the RNA binding proteins interact. So the RNA is like dynamics. It is needed to translocate from nucleus and the cytosol, and in this process, RNA binding protein is like a cargo to hold the RNA and transport it from different compartments of the cells. And also it stabilizes the RNA and it controls the translation, localization, modification, and the process of all the RNA process. And when we compare the structure of RNA binding proteins to the disease-related tau protein, and we found they have some of similarities. For example, the glycan-rich structure, the motif, and also the repeated domain motif. And when we look at the postmortem human brain tissue with the neurodegeneration, we found the tau protein localized to these RNA binding proteins. Once the tau aggregate, it will recruit and colocalize with the RNA binding proteins. So we thought these RNA binding proteins have this liquid phase separation and incorporate into the tau complex, and then they become aggregated together. This is not only we first look at Alzheimer's disease, but it's not a unique feature of Alzheimer's disease. Actually, it happens many other neurodegenerative diseases. This one shows the frontotemporal dementia. It is related to TIA RNA binding protein and TDP43 RNA binding protein, and also the FUS. So we, based on these findings in human tissue, we developed a mouse model. The mouse model developed the tau pathology, and we can follow dynamics because in humans, the only tissue we have is the post-mortem tissues. That is the very latest stage of the disease. But when we have the mice model, we can track the aging process from early stage to middle stage and later stage of the disease to see the development of the disease, and also check the dynamics of the protein interactions. So in this mouse model, we sacrificed the mice at 3, 6, 9 months. That is correlated to the early, middle, and later stage of the human brain, and try to find the colocalizations or interactions of these RNA binding proteins with tau pathology. Surprisingly, we found that the tau proteins, when they are small aggregates, the oligomers colocalize with RNA binding proteins. But once they become aggregated into fibrils, the large tangles, they dissociate with RNA binding proteins. That means the oligomers actually is the key, is the middle stage, is the most important part that later to cell death. These are some markers of RNA binding proteins, TIA, TDP, and G3BP. So based on that mice model, we can summarize that the protein aggregate and induce the translocation of tau proteins from axon into cytosol, and the tau proteins will recruit the RNA binding proteins from nucleus into cytosol. And when they recruit the RNA binding proteins, it will trap the proteins and form the complex. This complex will become accumulated more and more, and then they become irreversible and become a deposit in the cells. So that this deposit of the aggregates will disrupt the normal function of the neurons, and the neurons will die. Based on that, so what we can do to reverse the disease or stop the disease, and we now have a lot of targets and how we develop method to demonstrate we can help the disease and we can halt the disease progression. So in this mouse model, we knock out one of the RNA binding proteins called TIA. TIA is the key RNA binding proteins that are colocalized to tau pathology. So genetically, we remove the RNA binding protein, and we try to see whether this is helpful for the disease. So this curve shows the survival of the mice. The black part is with pathology, and the red means we knock out one allele of RNA binding proteins. It's really helpful. It extends the lifespan of the mice, and also make them the disease reduced, and the survival can be extended to more than half of the mice can more than 12 months. And if we remove all of the RNA binding proteins, it even helpful more. So that this study demonstrates our hypothesis that if we remove part of the RNA binding proteins, it's helpful to halt the protein aggregation and can rescue the mice from disease. And not only the survival, we also look at other behaviors of the mouse. For example, the white maze. The white maze is a test for the memory. So we can find that once we remove the TIA, the memory comes back. comes back to the normal, like the wild-type mice, the normal mice, and also the normal objective recognition. This is the behavior test for the cognitive decline. So when we have the disease progression, then the mice will progressively have the memory loss and a cognitive decline. But once we remove the TIA, the RNA-binding protein, the memory becomes better and the cognition becomes better. This is really surprising and really important for the field. And also we look at the pathology, we can clearly see once we remove part of the RNA-binding protein, the neurons become small. These black dots, that's the neurons. So in the normal conditions, they were condensed. This for a region called hippocampus. It is related to memory. And when we have pathology, the neurons get lost around 50%. But once we have the genetically knockdown of TIA, we have the recovery of neurons back to almost the normal condition. So this study give us the insight that we can focus on the RNA-binding proteins for the disease therapeutics development. So, but still, like I said, we don't know which step is most important, because for the genetically treatment, we remove the RNA-binding protein from the beginning. But in human, if we want to treat human, we can't remove that gene once we were born, right? So that is not feasible for reality, like how to treat the patient. So based on that scenario, we needed to figure out at which stage, the specific time point to make the treatment, like treatment on the animals, like the treatment in humans. So based on that, we have this hypothesis that is the tauoligomers, because our studies found tauoligomers are more toxic. Maybe we can focus on the tauoligomers. So once the oligomers started to aggregate, we disrupted the oligomers and stop the disease progression. That is the middle age of the disease. So that by that stage, we can we have diagnosis for the disease. So then we can stop it and reverse it. That's the right time point for the intervention in reality and for humans. So we have this study to compare the toxicity of oligomers and fibrils. We found that oligomers induce cell death, but fibrils didn't. So if we focus on the latest stage, that is too early, we have no way to recover the disease. So it's important to find the middle age events. So in this study, we inject the tauoligomers into the mouse brain, and then monitor the spreading of the aggregation and monitor the progression of the tau aggregation. And then we found that the oligomers are toxic. And so here shows the diagram how we did the experiment. We put the oligomers into the cortex. So this is the cortex, and then it goes to the hippocampus. It's loaded to the hippocampus, and then we monitor spreading on other brain regions based on the neuronal circuitry, including CA3 and internal cortex. And 3 months after the oligomer injection, we monitored the pathology, and we found 3 months later, oligomers induced a large amount of neuronal loss, about 50%, and accumulated of tau pathologies labeled by MC1, the red color. The more red means more pathology. So the S1P induced a lot of pathology in these mice. And once we have the TIA knockout, so we also can stop the spreading, stop the propagation of these tau oligomers. So this is another way to, compare to the genetically knockout, this is another way to demonstrate that we can stop the disease even it already started. We can stop it from the middle. And we can see once we stop the tau oligomer spreading, we can preserve the neurons, and it comes back to so we can reverse the 50% loss of the neurons. These are some other markers showing the neuronal loss. Now I needed to understand what other proteins we can target on. TIA is one of the proteins, but once we stop TIA, how about the complex? How about the dynamics of these protein interactions? So I designed an optogenetic tool. So this one, we can shine blue light to control the aggregation. So then we use immunoprecipitation to pull down the complex. Then we run the muscles back to find out all the proteins that is changed in the early stage, in the middle stage, and later stage. So we can control the disease progression in an integrated way, not only on one gene or on one protein. We can control all the proteins, all the pathologies, and make the neurons healthier. So this is how this video shows how it works. This video shows how the proteins aggregate in live cells, like the dynamics of protein interactions. And in this method, by this method, we found a very important study. We are the first one found this. So the tau proteins interact with a protein called HRNP-A2B1. These proteins are RNA-binding protein, usually located in the nucleus, but in Alzheimer's disease, it highly redistributed. It distributed in cytosol and colocalized to the tau aggregates. And in these tau aggregates, not only HRNP-A2B1, we found the RNA is trapped here. The RNA are methylated by the methyladenosine. So that is the M6A. So now in the beginning, we found the protein-protein interactions, but on this step, we found not only proteins are interacting, but also the RNAs. The RNAs were trapped, and the RNA modification was changed. RNA modification is related to protein translation, the generation of new proteins, and also the protein degradation. That means if once the RNA modification is changed, the entire RNA metabolism is disrupted. All the proteins cannot be translated because we always need new proteins to be synthesized. Now, by this method, in this study, we found, the actually protein synthesis is stopped.
So can I just ask you a question? So when you're talking about the TIA1 knockout, right, to stop the spread of tau oligomers and stop the disease, and you're seeing reversal in 50% loss of the neurons, right? So what happens when the neurons come sort of back online? Are you saying that this stress granules disappear and messenger RNA can go back to its original functionality? Are you saying that maybe the cells are senescent and they in some way come back online? I mean, what do you think is the mechanism of how that works?
That's a really good question. Yes, we look at the pathology. Once we remove TIA, the pathology disappear, not totally disappear, in some degree disappear. So the aggregation become less, neurons become healthier. And we also look at the behavior. The behavior become better. That means we slow down the disease, but we can't see we totally remove the disease progression, but we slow it down. The mice survive longer. So that is really helpful.
And do you think it's due to the fact that these stress granules are disappearing, the ones that are currently there, or that no new ones are forming?
We think that is no more new ones are forming. And also we monitor the protein synthesis. We use a puromycin. It's like amino acid, so incorporate into the newly synthesized proteins. And we found the protein synthesis comes back. Because once stress granules forms, the protein translation and synthesis is stopped and reduced. Once we have the RNA protein knockout, the protein synthesis comes back. So the neurons have more normal function and a better performance.
Is there a half-life of the stress granule?
Yes. The stress granules have different, like I said, have acute stress granules and also chronic stress granules. the acute stress granules usually can disappear in 3 hours. But the chronic one is persistent. It will never disappear. Yeah.
Okay. Yeah. Okay. Sorry. I just wanted to kind of clarify a couple of those things. It's fascinating.
Yeah. And also in this study, we found a key mediator, the hRNP-A2B1. TIA is one of the key mediator for tau pathology. And hRNP-A2B1 is a key mediator specifically to oligomers. I introduced oligomers is the key tau species related to cell death and cytotoxicity. And based on that study, now we know, oh, we need to remove the modification of RNA. We need to reduce the hRNP-A2B1 to stop the disease from early stage, from when the oligomers forms, before neurofibrillary tangles forms. We needed to stop the disease progression from early stage, and then we can reverse the disease and become more powerful therapeutics. So we tried that in mice. In my publication in this paper, the molecular cell paper published last year, we tried in the tauopathy mouse models, in cell lines, and also in primary neurons. It's really helpful if we remove the hRNP-A2B1. It's protective to the neurons, pathology, and behavior. But as it is similar to TIA, it's partially protective. It's not like completely remove the pathology. And based on that, we have another strategy is to control the RNA modification, because RNA modification actually is the real mediator to protein synthesis and to the protein degradation. So we also wanted to test it on the human cells. So we developed this 3D brain organoid from the AD patient iPSC. So we have the iPSC derived from the AD patient, and then developed into neurons, astrocyte, and microglia, the 3 main cell types in the human brain. And then we developed a strategy to force the assembloid of these 3 cell types and make them form a brain-like organoid. Then we tested the therapeutic strategies on this brain organoid. And we used the single-cell RNA-seq to find out the interaction with different cell populations and the cell response to the treatment. And we found that the RNA metabolism is highly changed in disease condition. And when we have the treatment, the RNA metabolism can be rescued. And this picture shows how it works. So this M6A, that's one of the RNA modification. It is highly increased in the diseased conditions. And the TOMA2 is the tau pathology. It also accumulated, and neurons get 50% loss. And CD11B is the microglia marker. That is the inflammatory response. We can see the microglia is activated, and we detect the neuroinflammatory factors. But when we have the treatment of M6A, when we remove the the excess of RNA modification, we rescue the neurons. M6A reduced, pathology reduced, and neurons become healthier, and the organoid become more integrated. This is some quantification of the pathology and the neural markers. So now we summarize our findings. So we try to find the fundamental mechanisms leading to neuronal death, neurodegeneration in neurodegenerative diseases. And we found stress granules is the key component. It attracts the misfolding proteins, it attracts RNA-binding proteins, and also we found the RNAs is also recruited and become modified. So based on that findings, we now have this focus on the central biochemical pathways to treat the disease. We knock down the key RNA-binding proteins, we remove excessive RNA modifications, and we can protect the neurons against pathology and make the behavior better and help neurons healthier. So how is it linked to diabetes? Actually, the etiology of most of neurodegenerative diseases are unclear. Environment interacts with the genetics to initiate the disease. We know the tauopathy, all those cellular mechanisms. That's just what we found happened in the cell, but we don't know what caused these events. So other disease, for example, cardiovascular disease, diabetes, diet, those are all risk factors of neurodegeneration. The diabetes is also highly linked to the RNA metabolism. So because in our study we have this normal finding, the RNA metabolism is disrupted in neurons. So when we have the RNA metabolism in other cell types in the thalamus or other brain regions, that may control the diabetes happening. And also the different diet and also the risk factor, other risk factors to diabetes and also genetic changes also related to the neurodegeneration, because the nervous system controls all these the peripheral systems. So I think that's important to study the connections between diabetes and neurodegeneration. Actually, in our lab, we always talk about this, like how the vascular structure influence neurons and how the metabolism influence the neurodegeneration. So this is a good opportunity to think the disease progression in a more broad view.
Yeah, no, it's very interesting to think about the interaction of physiological systems, right?
Yeah.
Which is exactly what you're talking about.
Right. Yeah, that's all our new findings. I hope we someday can have some clinical trials with these new drugs we just found. Especially this drug already FDA approved for cancer disease. So we thought, oh, this is really important. Now we use a different dose. We can treat neurodegeneration. It's safe, so it's kind of more promising compared to other newly found medicines. Yeah. Yeah. And last, I would like to thank all our collaborators, our lab members, and the fundings from NIH and also from different foundations, Alzheimer's Association, Brady Folks Foundation, and also the Kelly Chan Fund. These are all great support to our. Without this support, we can't finish this research and we can't go more for these studies. Yeah.
Yeah, and I think that, you know, this is a very interesting, you know, it's an interdisciplinary, you know, talk. It's not directly focused on type 1 diabetes, but, you know, there's been a lot of literature, a lot in the literature, you know, basically implicating whether or not stress granules may be, you know, stress granule formation attenuates RAC1 mediated apoptotic cell death induced by Marusin. That paper came out in 2020. And as we talked about, the tau ablation in mice led to pancreatic beta cell dysfunction and glucose intolerance. That paper came out in 2018. Decio Zurich has talked about sort of what are the molecular footprints, and also about, you know, what makes beta cells a vulnerable autoimmune target in type 1 diabetes. And perhaps these kinds of mechanisms, you know, it might be great to have a little more attention focused on them to see, you know, what is actually happening in context of, you know, the beta cell, you know, tau and other and the stress granule situation. And whether or not that plays a role in, you know, as they become stressed, you know, do they have some of the similar, you know, mechanisms that are occurring in neurons, and could those be addressed in similar ways? So I think that's where, you know, the overlap happens, and I think your work is really interesting and exciting.
Yeah. Yeah, that's important. And also we think that one of the key proteins in diabetes is called presilin.
Yes.
Yeah, presilin is it has been well studied in Alzheimer's disease. It is a key protein in the protein aggregation. It's also in the neurons with the tauopathy. So that's also that's one of the interactions between diabetes to AD and other dementia.
Yeah, and I mean the beta cell, is a very neuronal style cell, at least operationally. So it's always important to kind of like see what's happening in these other disease states and see what can be gleaned or learned to bring into type 1 diabetes. I would like to thank you very much, and you went a little over. I appreciate that because of the questions. But Dr. Jiang, thank you again for talking to us. And I encourage anyone who is listening to this who has further questions to reach out to you. And who knows, maybe it'll spark a collaboration.
Yeah, thank you very much, Monica.
Thank you. Have a great rest of the day.